Assalamualaikum everyone. So in this video, we are going to continue with part 2 of chapter 8. And previously we have already looked at the different types of blood vessels in the mammalian transport system. And in this video, we are going to discuss some other components of the mammalian transport system. Before we move on further, there is something that I would like to show you, which I forgot to show it in the previous video. This is a chart that shows how blood pressure changes as the blood travels in one complete journey from the heart through the systemic circulation, back to the heart, and then through the pulmonary circulation. As you can see, the pressure will be the highest for arteries closest to the heart, as they will be the first to receive the surge of blood from the heart. And between systemic circulation and pulmonary circulation will have lower blood pressure. And this is because the right ventricle, which is responsible in producing the pressure in the pulmonary circulation, have much thinner walls, because it only needs to push the blood to the lungs, right? And as you can see in the diagram, there are fluctuations throughout the chart, right? You can see it here, right? It goes up and down throughout the chart. And this shows that blood moves in pulses. The higher peaks that you see along the line symbolizes the blood pressure produced when ventricles contract. And the lower peak that you see here, it symbolizes the blood pressure produced when ventricles relax. And since capillaries and veins are located further away from the heart, and the blood flow is quite slow, the blood pressure for both of the vessels, the veins and capillaries, are very low, right? And you can't really see any fluctuations there. It's just a straight line, through the capillaries and through the veins. The diagram that you see here is a diagram that shows how blood pressure is measured. What is the actual concept behind the measurement? And by the way, the equipment that you see here is known as the sphygmomanometer. Okay, we have a few units in the biolab, and inshallah, once the CMCO is over, okay, you can try it yourself. Okay, the working principle behind the sphygmomanometer is quite easy. It's quite simple. First, you need to put an inflatable cuff around the arm. Okay, usually the left arm will be used. It is more appropriate as the artery that runs in the left arm is closer to the heart and aorta. Once the cuff is secured around the arm, it will then be inflated. Okay, this is done to squeeze the underlying arteries and cut off the blood flow. Okay, you will know that the blood flow has been totally cut off when there is no more pulse sound, heard through the stethoscope. Okay, so if you put a stethoscope just underneath the cuff, once it has been inflated, when you can no longer hear the sound of the pulse, it means that the blood flow has been totally cut off by the inflated cuff. Okay, usually there is a pressure sensor in the cuff, right? And the reading at this particular time, okay, when the blood flow has been totally cut off, in the arm would usually be more than 120 millimeter mercury. The cuff will then be slowly deflated, allowing blood to flow back to the left arm or to the right arm, whichever arm that you use. And the first sound that you hear through the stethoscope will be the first pulse of blood passing through, right? The sound of the first pulse is the systolic reading or the systolic pressure, right? The upper reading of the blood pressure, and usually will be around 120 millimeter mercury, okay, when checked with the pressure reading on the sphygmomanometer. And as the cuff deflates even further, okay, there will be no more pressure given to the underlying artery, and the final sound of pulse, okay, that you hear through the stethoscope will be the diastolic reading or the lower reading, okay, which is usually around 80 millimeter mercury. The unit of pressure in the sphygmomanometer is given as millimeter mercury. So normal blood pressure, the average normal blood pressure is usually around 120 for the upper reading and 80 for the lower reading. Okay, so let's move to the next component of the mammalian transport system, which is the blood plasma and the tissue fluid. So what is blood plasma? If you look at the diagram that we have here, basically blood plasma is the fluid part of the mammalian blood. It helps to move the blood cells, such as the RBC, okay, the red blood cells, the white blood cells, and platelets in the blood vessels. So without blood plasma, it is almost impossible to move those cells inside the blood vessels. The main component of blood plasma is obviously water, but there are also a lot of different solutes that can be found dissolved in the blood plasma, such as glucose, amino acids, lipids, protein plasma. So protein molecules that are found in blood plasma is known as protein plasma. You can also, find a lot of mineral ions, okay, and etc. And if we are able to draw out some of the mammalian blood, okay, and put it in a test tube, okay, put it in a centrifuge machine, spin it at a very high speed, okay, the blood will actually be separated into 3 different layers. Okay. If you look at this diagram, you can see that the first layer is the layer of plasma, okay, which takes up about 55% of the total blood. Okay, and then you have, okay, one thin layer in the middle, okay, that would be the layer containing the white blood cell and platelets, and usually it will be less than 1% of the total blood. Okay, and the layer at the bottom would be the layer for the RBC, okay, and it takes up about 45% of the total blood. Okay, so that would be components inside the blood: blood plasma, red blood cells, white blood cells, and platelets. During the circulation of the blood in the body, plasma will leak out from capillary to the spaces between cells. And this is due to the fact that there are a lot of small gaps on the endothelial layer of the blood capillaries. Okay? And once the plasma leaked out and bathed the surrounding cells, it will no longer be known as blood plasma, but they will be known as tissue fluid or interstitial fluid instead. Okay, and the tissue fluid or the interstitial fluid forms the immediate environment of each individual body cells. Okay? Tissue fluid is almost identical in terms of composition to the blood plasma, right? They have very similar content. But if you talk about tissue fluid, it contains far less protein molecules than blood plasma, as most protein molecules are too large to pass through the capillaries. Right? So whenever blood runs from the artery to the capillary, most of the blood plasma components tend to leak out except the larger protein molecule. Okay, the volume of fluid which leave the capillary to form tissue fluid or the interstitial fluid is the result of two opposing pressure. Okay, if you look at the diagram, okay, the blood pressure at the arterial end of a capillary is high enough to push the fluid out, right? However, once the tissue fluid is out of the capillaries, okay, the tissue fluid lacks the high concentration of protein that exists in plasma. Remember I said just now, okay, everything can actually leak out from the capillary, okay, into the surrounding cells except protein molecule. Right? So tissue fluid lacks the high concentration of protein. And in other words, we can say that tissue fluid has higher water potential, okay, than the capillaries. And this will cause tissue fluid to move back into the capillaries, especially near the venous end. Right? So those tissue fluid that has already moved out, okay, tend to move back into the blood capillary at the venous end. However, the fact is that there are more fluid flowing out at the arterial end rather than those that move in at the venous end. And we are going to discuss later on how this excess fluid can be returned back to the blood vessel. So these are some of the points that I have already discussed just now. Okay, so next we are going to look at the formation of the lymph. Remember I said just now that in every circulation of blood in our body, more fluid will leak out in the capillary compared to fluid moving in. So it means that whenever blood passes through the capillaries, we are losing more and more body fluid. Right? So there should be some sort of mechanism to take back those fluid and return them back to the blood vessel. It is estimated that only about 90% of fluid that leaks from capillaries will eventually seep back into them. Right? So only 90% that leak out, okay, is taken back by the capillaries. So you have 10% more that will not enter the capillaries back. So what happened to that 10%? So that 10% tissue fluid, okay, will be collected up and returned to the blood system or returned to the blood vessel by means of series of tubes known as lymph vessel or lymphatics. Okay, so what are lymph vessels or lymphatics? They are basically tiny blind ending vessels which are usually found side by side with the blood vessels in almost all tissues of the body. Okay, so lymphatic vessels contain tiny valves which allow the tissue fluid to flow in but stop it from leaking out again. And the valve also allow large protein molecule to pass through. And this is important because this controls the osmotic pressure of fluid between the cells. Because too many protein in the tissue fluid will cause water to move out from the cells, and this might damage the cells. And once the tissue, once the tissue fluid is inside the lymph vessel, it is known as lymph, right? So once it leak out from the capillary in every turn of blood circulation, okay, they become tissue fluid. But once the tissue fluid move into the lymph vessel, they will be known as lymph. Some points on lymph formation that we have discussed just now. If you look in the diagram here, you can see that smaller lymph vessels join up to form larger lymph vessels. Okay, they are just like blood vessels, right? You have the larger one, the aorta, the arteries, and also the capillaries. And remember that the function of the lymph vessel is to return the 10% fluid, the 10% tissue fluid that has leaked out from the capillary back to the blood vessel. So if you look at the diagram again, you can see that these large lymph vessels will join up with the blood vessel at certain location in the human body in order to return back the 10% tissue fluid that failed to be taken back by the capillaries. Okay, and if you look at this diagram, two major lymph vessels are the right lymphatic duct, the one that is shown here, and the thoracic duct. Okay, you can see it here. Right lymphatic duct will pick up tissue fluid from the upper right part of the body and drain them into the right subclavian vein. Right, so right subclavian vein are the blood vessels that join up with the lymph vessel, okay, of the right lymphatic duct. While thoracic duct will pick up tissue fluid from the left side and lower part of the body to be drained at the left subclavian vein. Right? So there are 2 points where those lymph are drained back to the blood vessels. Right? First one is between right lymphatic duct with right subclavian vein, and the other. Next, we are going to look at some of the types of blood cells that can be found inside the blood vessels. And those cells are suspended in the blood plasma. Okay, total volume of blood inside the human body in average is roughly about 5 decimeter cube, okay, or 5 liters. And in terms of blood cell components, there are roughly about 25 trillion RBC, or red blood cell, 500 billion of white blood cells, and about 6 trillion of platelet in average in a human body. So this is a photomicrograph of human blood that has been stained. Okay, and the staining allows you to actually see the differences in terms of structure of those blood cells. Right, you can see the red blood cells. A huge amount of the cells are red blood cells over here. Okay, and there are a few different types of white blood cells as well. Okay, here, if I'm not mistaken, the one with the large nucleus here, this is the monocyte. Okay, the smaller one over here is lymphocyte. Okay, and the one with the earlobe kind of nucleus is the neutrophil. Okay. So let's look at the first blood cells, which is the red blood cells or the erythrocytes. Their red color appearance is caused by the pigment known as hemoglobin. And the main function of the red blood cells is to transport oxygen from lungs to respiring cells or tissues. Red blood cells are produced by the bone marrow, and they have a very short lifespan of about 4 months or roughly 120 days. In terms of structure, red blood cells are shaped like a biconcave disc shape. This is to increase the surface area in relation to its volume. And having this increases the efficiency of oxygen to diffuse into the RBC. Red blood cells are also very small and flexible. The diameter of red blood cells is about 7 micrometer. And this feature is important as most capillaries have slightly larger diameter than that. And some capillaries might even have slightly smaller size than 7 micrometer. And being flexible means that red blood cells can still squeeze themselves inside these capillaries, even though they are relatively smaller, right? And red blood cells do not have nucleus, they do not have mitochondria or even the endoplasmic reticulum. Having less organelles means that they can fit in more hemoglobin to maximize the amount of oxygen that they can carry. Some description on red blood cells. You can copy this later. The next blood cell is the white blood cells, or sometimes referred to as the leukocytes. They are also made in the bone marrow, but they have even shorter lifespan, okay, of about 35 to 42 days as compared to the red blood cells. In terms of structure, okay, all white blood cells have a nucleus and they are larger in size compared to the red blood cells. Okay, some white blood cells have irregular shape, while some might have spherical kind of shape. And in terms of function, okay, we can divide the white blood cells into two different types, which are the phagocytes, okay, and the lymphocytes. Okay, phagocytes are cells that destroy invading microorganisms by engulfing them in a process known as phagocytosis, while lymphocytes are cells that destroy microorganisms by secreting a chemical known as antibodies. Okay, and we are also going to learn this in detail in chapter 11. Okay, so I guess I'll stop here, okay, for now, and we are going to continue the third part of chapter 8 in the upcoming video. Okay, so with that, Assalamualaikum warahmatullahi wabarakatuh.